yesterday. I'm your chairman for this session and the session afterwards, which will be both on cancer nanomedicine. My name is Jan Mollenhauer, and I'm from the University of Southern Denmark. So our session will be about targeted and personalized cancer nanomedicine, and it's my pleasure to welcome the first speaker, Professor Kostas Kostarelos from the School of Pharmacy at the University College London, and he will talk about looking and listening to light from liposome nanostructures for cancer theranostics.
Thank you.
So thanks for coming, and we are curious.
Good morning, everyone. I have to wake you all up. That's my task. Beat told me that you have to skip dinner and come here at 8 and get everybody up to their feet. Thank you for your courageousness to wake up at 6:30 and come here at 8 to listen to me talking to you about liposomes. The game is not fair. You already started counting down before I started. So I will talk. I'm always trying to bring new work from our laboratory here in this meeting. So true to form, I'm trying to show you some of our recent data that it has to do with liposomes this time, and not carbon nanostructures as we usually bring in this meeting. And I'm assuming that a lot of you, probably all of you in this conference, appreciate liposomes because they are perhaps one of the most clinically, perhaps the most clinically established delivery system. And apart from the clinical profile that they also offer a very interesting modular capability for engineering new structures. So apart from what we know that liposomes can do, what we are trying to do, and what I'm going to share some new data with you this morning, would be about what is new, what is new about liposomes, and what new thing can we achieve by re-engineering liposomes. So a few years ago, we tried to create new structures using the liposomal bilayer as what I call a nanomedicine platform. So that's a slide that summarizes work of about 10 years. So we put metallic nanoparticles in liposomes. We put fullerenes in liposomes, both in the aqueous space and in the bilayer. Quantum dots, and different types of viruses for gene transfer applications. So these are actually relatively simple structures because a couple of years ago we proposed liposomes as a theranostic template. And that is one step higher in terms of complexity because you're talking about two functions here, a therapeutic function, which is shown on the left-hand side of the slide, and a diagnostic function. And in this paper, what we proposed, based on our published work and other people's published work, is that you can build those kind of more complex structures in order to build theranostic agents. So in this schematic, you see doxorubicin quantum dot liposome, or this one, you are looking at the optically tagged adenoviral particle wrapped up in a liposome. So these are more complex structures. What I will show today is two case studies along the same lines of building new structures that will contain two types of functions, a therapeutic function and a diagnostic function. So the first case study I will talk about, and I'm trying to keep consistent with the title of the presentation, and I'll explain to you. I hope it will be very clear what do I mean by saying looking and listening at those liposomes. So in this particular case study, we're designing a rather complex structure, which is made of cationic liposomes, gold nanorods, and siRNA. So the hypothesis behind this concept is that we can engineer cationic liposomes. A lot of people have done that. They've been through a lot of clinical trials, so there's not anything new in this respect. And we are building gold nanorods on the structure in order to design a new type of positively charged vesicle, and then we're complexing that with siRNA. The second part of our hypothesis in the design of the system is that we can listen to those structures and listen to their optoacoustic signal, and I will give you a little bit more detail about that in a minute. And at the same time, try to look at the fluorescent signal by either adding an NIR, a near-infrared probe, on one of the lipids we are using to build the liposome with, or if we fluorescently tag the siRNA. And the whole reason behind doing that is to be able, or attempt to try to track both the therapeutic agent, the siRNA in this case, and the vesicle once they are localized within a tumor. So the first question that we are asking, or I would like to explain to you, is why use nanorods? I think that those of you familiar with this field have read or came across this kind of concept, that nanorod will consist of their longitudinal axis that will offer a modular capability to push the spectrum that you're picking up absorbance from towards the near-infrared region. So the ones that I'm going to show you the rest of the data from is these ones, the ones in the middle. So these are 38 nanometer long nanorods, gold nanorods, that we will place on a bilayer, and we will read their absorbance, as you will see in a minute. So this is a very standard procedure by which we're building liposomes with. So this is the field of hydration. So you're dispersing your lipids, in this particular case DOTAP cholesterol, in a solvent phase. You're evaporating it, you're forming a film, you're rehydrating the film in the presence of your gold nanorods. So you're building this kind of structure in schematic. I'll show you in a minute what's the reality. And here you also have the possibility to build this bilayer using another type of lipid, which will contain a second infrared probe. So this is how the structures look like under electron microscopy. This is without the nanorods, and this is with the nanorods, and you'll see how the nanorods are positioning themselves very nicely, very beautifully around the bilayer. And this is how the AFM image looks like. If I zoom in, you'll see again the very fine arrangement of the gold nanorods around the lipid bilayer. We can discuss later on why and how these structures are put together and why they're stable. I don't have time to go deeper into the physical chemical interactions that are very interesting between the nanorod and the lipid bilayers. But these are solid, robust, stable systems, both in serum and in the body of the animals, as you will see in a minute. But what is very interesting from the concept of what I'm trying to put across to you this morning is the optical properties of the systems, because we have the absorbance, as I mentioned, you offered by the nanorods, and this is the signal that we're going to listen to. And at the same time, the same construct, because of the NIR probes that we build the liposome with, we also have a fluorescent signal, and this is the signal that we're looking at. So what we're going to do, as you will see in a minute, we're trying to correlate the two signals and co-register both signals, the same liposomes. How do we do that? So how do we listen to light coming out of those liposomes? We're using this I mean, if you talk to it depends whom you're talking to. If you talk to the optics community, they'll tell you it's well established. If you're talking to the biomedical, nanomedical community, they'll tell you it's fancy. So it depends whom you're talking to. This is an optoacoustic tomography system that we're very strongly collaborate at the Helmholtz Institute in Munich with Professor Giachristo's laboratory. And the system, the concept is that you're putting the animal here in this bed, and you're irradiating it with a laser beam, and then you're picking up the optoacoustic signal that's coming out of the probes that you have there. So you're registering the optoacoustic signal, basically the ultrasound that's coming back from the animal, and you're building an optoacoustic image. So you will see what kind of images we are able to obtain, and I think you will appreciate why this is interesting. So we're following standard procedures, very straightforward. This is an intratumoral injection of those liposomes, and we're using an intratumoral injection because we're talking about cationic liposomes, so they wouldn't have much fate if you were injecting them intravenously. In this particular case I'm showing you. And this is how the animal is positioned. The xenograft is built at the back of the animal. We're injecting into the tumor, and these are the kind of landscape of the images you're going to see in a minute. So this is how the images that we're listening to, the optoacoustic signal, looks like. This is the tumor after 1 hour and 6 hours. and that's the tumor, that's the body of the animal. And this is what we co-register, this is what we're looking at, if we're looking at the fluorescent signal. So we sacrifice the animal, cryosection the whole animal, and you see again, and this is the signal that's coming from the NIR probe. What is interesting about this that I want to focus your attention to is the detail that we're getting in a non-invasive manner by optical methodologies, which is very interesting. One step further in terms of complexity, we add siRNA. The particular example I'm showing you, we're adding siRNA, which is tagged with an NIR probe as well. So this is the signal we're registering from the nanorods, the listening signal, the optoacoustic signal. So after 10 minutes, this is how they look like into the tumor. After 24 hours, you'll see again the spread within the tumor. And this is how the co-registration of the siRNA signal looks like. So we know where the liposomes, where the siRNA is. And if you want to get fancy and look at the biological activity, this is what you get when you're sacrificing the animal, sectioning the tumor, and looking at the apoptosis that the siRNA is able to induce after gene silencing. So we know that the siRNA we are injecting into the tumor works. And this is with thick sections and this is with thin sections. So we're definitely getting apoptosis inside the tumor. Therefore, the siRNA works. I'm not going to spend too much time longer on this. I just want to show you the concept of what kind of complex liposomes you can build with and how you can track 2-3 signals at the same time. The second case study, and last in this talk, would be about an alternative system that will be trying to look and listen at liposome ICG vesicles. And the hypothesis behind the work is that we can engineer a pegylated system, a pegylated liposome. This is standard doxyl type of formulations with an indocyanine green containing probe in the bilayer. And there are a few papers, not a lot of papers in the literature that are talking about those systems. If you go and look a little bit carefully about this, this is not really established technology. So we spend a lot of time trying to optimize this system. It's not a straightforward system. So what we're doing here, what's different is that we have a pegylated system, therefore we can inject intravenously in this case. And what we're trying to do is listen to the optoacoustic signal coming from the ICG. But also at the same time, what's beautiful about the system is that the ICG offers a fluorescent signal. So we're trying to simultaneously listen and look at the signal from the signals offered by the ICG and perform dynamic tumor localization studies by injecting intravenously those constructs. Why ICG? I'll go very quickly here. For those of you that are not familiar, this is perhaps, I believe it's the only optical probe that has been approved by the FDA and it's used for a variety of clinical applications that you see here. This is the structure, but what is interesting from the optical point of view is this spectrum here. So this is the absorbance spectrum of some biological relevance. This is the blood signals and this is the water absorbance signal. And this is where ICG lies. And these are the two signals that we are trying to collect from these molecules. So as you can appreciate, what we're trying to do is listen again at the absorbance of ICG at 780, and at the same time look at the fluorescence offered, so the emission at 820. Same molecule, same vesicle. This is how those vesicles look like, about 100 nanometers in between 100 and 150 nanometers in diameter. And the ICG, as I said, this slide is not doing any favor to our postdoctoral fellow who spent at least 6 to 8 months optimizing the system. So it looks very simple and straightforward. It is not. In any case, what you see here is that if you're doing an absorbance stability study in days, you'll see that if you put ICG at this concentration on liposomes, you have basically a very stable system, but the ICG alone drops after 1 day. These kinetics are much worse when you're putting these objects or molecules into blood. So how do we do our imaging? The first thing we did is we looked at the signal. So when I see look, we're doing IVIS. IVIS is a very standard, well-established preclinical imaging modality. We're injecting intravenously here. The animals that I'm going to show you do not contain any tumors. We're picking up the signals, and I'll show you why IVIS is beautiful because of the simplicity of the concept and the methodology, but it's very restrictive because these are the kind of images that you see when you're performing those kind of experiments. This is the ICG alone injected in those animals at 5 minutes, 1 hour, up to 24 hours. So you see basically nothing after 24 hours. And if you kill the animal and pick up the organs, you'll see really nothing. Whereas with the liposome, you start seeing those very broad signals. But again, you see nothing after 24 hours. But if you open up the animal, as you would expect, you see a very strong liver signal. And this is where optoacoustic tomography is becoming also very interesting. We are injecting intravenously, and now we're listening at the signal from the ICG. So we're doing optoacoustic signal registration. This is how the tumor and the animal looks like. So the tumor is here, the body of the animal is here. And 5 minutes after injection, you start seeing signal in the vasculature. After 1 hour, you start getting the signal, the liposomes coming into the tumor, and you'll see very beautiful images after 24 hours as the liposomes have extravasated. That's with one type of tumor, the 4T1 tumor. If you do the same experiment with a different type of tumor, and this I think where the clinical applicability and relevance of these systems is lying, you'll see that you have, even after 24 hours, almost nothing in the tumor. So there's very poor extravasation with this tumor. So there is a tumor-to-tumor variability, as we all know, and you can see it very, very clearly with these optical imaging modalities. Now you can go fancier and use more experimental techniques and go high resolution optoacoustic perform optoacoustic signals at high resolution. So again, intravenously inject the liposomes. Here's the tumor in the animal. Again, we're talking about non-invasive techniques. The animal is anesthetized, is alive. You can wake it up and play games with it. So what you're doing is, as the animal is anesthetized, you're flipping it over and you're taking two registrations at two planes, as you can see here, plane 1 and plane 2. And if you do high resolution, you start seeing beautiful images of liposomes in the tumor vasculature or extravasating. I'm almost there. And then if you do many planes, if you start registering many planes, you can build three-dimensional images of how the vasculature of the tumor and how the extravasation is taking place as we go along. And you can, all of this is done with optoacoustic tomography. And then in order to validate your results, that's the beauty of ICG. You can look at the signal, so the fluorescent signal, by picking up this tumor. So what we did is here is we took the tumor, and the data I'm going to show you is only with the tumor, not the animal is dead by that time. And you can section it, cryosection it, and build multiple plane imaging, as you can see here, the sagittal and the coronal sections, and you can reconstruct and therefore register in 3 dimensions where and how the tumor is spreading. So you can validate your imaging, your non-invasive imaging, post-mortem. And this is how it looks like in the actual cryostat, whereas you're collecting the signal. This is the histological image, this is the tumor, and this is the signal from the ICG liposomes. So to conclude, I showed you some previously unreported liposome nanorod hybrids and liposome ICG systems that have been successfully engineered based on a redesign principle, and the strong belief we have in our lab that liposomes are not boring, they're not dead. We can do very fancy, very interesting things with liposomes. And the liposome nanorod and ICG systems that I showed you can be resolved in vivo deep within the tissue using optoacoustic tomography or other techniques. Optoacoustic tomography is our modality of preference because of the simplicity, of course, and both in terms of chemistry and in terms of pharmacokinetic behavior of the systems. And the last point I want to make is that these novel liposomes show high optical and structure stability in vivo, as you have seen, and we can monitor very nicely the kinetics of the liposomal systems by looking at a variety of different signals. And again, our kind of dogma here that we can build novel liposomal structures carrying both therapeutic agents and imaging modalities to constitute new platforms and new theranostic agents. Our funding from the Commission and various UK-based funding agencies in collaboration with AstraZeneca I need to thank the postdoctoral fellow in our lab, Dr. Neus Lozano, who did most of the work I showed you on these liposome structures, and our guys in Munich, Vasilis, Nicolas, and Antonio, that performed a lot of the imaging that I showed you with us. Thank you.
Yeah, thanks a lot, Kostas. It was very impressive looking and listening to you. Other questions from the audience?
Hello. Thank you. I agree that the ICG is a very good fluorescence dye, and your results are very convincing. So my question is, how well ICG is retained in lipid membrane during circulation, and can we use this ICG-loaded liposome to quantitate the total amount of liposomes accumulating in tumor tissues?
Thank you. Yeah, the second part is tricky. The first part is straightforward. It's very stable. We've performed very long-term studies in full blood. There's no problem. I mean, in terms of liposomal pharmacokinetics, we're talking about a window of 2 days, 48 hours. Everything has happened within this time window. So the system is very robust for this, in the context of this time frame. The second part of your question is very tricky, and I know that the guys in Munich are very interested in. I do not believe that we can do quantitative pharmacokinetic analysis with ICG liposomes yet. You can attempt it, but it's not going to be reliable. So, yeah, if you're courageous enough to go ahead with it, I would suggest you also run in parallel a study with radio-labeled lipids, just to be on the safe side, just to correlate. But it's, I wouldn't do it.
Okay, time for one short question, a short answer.
Yeah, how close is this to being ready for any human?
Yeah, well, Vasilis and I wanted to be very ready. I don't know how the clinicians... look, there's nothing new about this. That's the beauty of this, especially with the ICG study. And this is why we both believe that we can relatively quickly translate that clinically for applications like intraoperative applications, for example, surgical applications. I also would like to see those systems for screening, as a pre-screening tool for different patient tumors, to see how vascularized people's tumors are. I'm not sure how easy is it. It's not my expertise, but what I know from my point of view is that we're using Doxil and we're using ICG, and we're not covalently conjugating them, so we do not contain any new moiety in this system.
Okay, then thank you again. And it's my